In what physicists call panspermia, the hypothesis that life's chemical ingredients rode to Earth aboard asteroids and comets, two pristine samples returned from deep space in 2020 and 2023 contained something no one expected. Sitting inert inside rubble pile asteroids that had never known liquid water or warmth was the complete alphabet of DNA and RNA, letter for letter. Adenine, guanine, cytosine, thymine, uracil. All 5 nucleobases confirmed in the 5.4 grams returned from asteroid Ryugu and the 121.6 grams brought back from Bennu. Less combined mass than a small apple, yet these grains carried the genetic vocabulary of every organism that has ever lived on Earth. The same molecules that spell out your genome were forged in the cold vacuum of space, riding dormant through billions of years, waiting. Stay with me, and you will understand why that delivery, extraordinary as it is, cannot explain the origin of life itself. You will see why the 700 million year silence between Earth's habitability and the first living cells points not to a problem with panspermia, but to a deeper question the hypothesis was never designed to answer. And you will learn why the evidence now forces a stark conclusion. Space wrote the alphabet, but Earth composed the first sentence. By the time the screen goes dark, you will know what pristine asteroid samples revealed about life's precursors that 50 years of meteorite studies could not confirm. Why NASA's lead scientist called one result from the Bennu sample analysis a finding that invalidated 20 years of research. Which competing terrestrial engines the evidence points toward? Alkaline hydrothermal vents, or volcanic hot springs, as the spark that activated an otherwise inert cosmic inventory. And where the science reaches its honest limit and leaves one question genuinely open. If you find value in explorations like this, the subscribe button quietly helps these deep dives reach more curious minds. So, here is where it begins. Panspermia, from the Greek meaning seeds everywhere, proposes that life, or its chemical building blocks, originated beyond Earth and arrived here through space. The hypothesis spans a spectrum. At one end sits radiopanspermia, the 1903 proposal by Swedish chemist Svante Arrhenius that bacterial spores could be driven between star systems by the pressure of starlight itself. At the other end sits lithopanspermia, the more modest claim that organic molecules, amino acids, nucleobases, sugars, could survive embedded in meteorites and seed a sterile planet with chemistry, not life. Between them lies directed panspermia, the 1973 speculation by Francis Crick and Leslie Orgel that an advanced civilization might deliberately scatter life across the galaxy, a notion that remains unfalsifiable and sits at the fringe of serious consideration. What unites all versions is the recognition that Earth is not isolated. 4 billion years ago, when the planet's crust was still cooling and oceans were forming, space was not empty. It was raining organics. The scale of that rain is measurable. Present-day Earth accretes roughly 40,000 metric tons of extraterrestrial material per year, most of it in the form of micrometeorites, dust-sized particles 50 to 200 micrometers across that survive atmospheric entry and settle onto the surface. The figure comes from satellite impact crater counts and ice sheet recovery missions. Love and Brownlee, working with debris captured by NASA's Long-Duration Exposure Facility satellite in low Earth orbit between 330 and 480 kilometers altitude, measured roughly 109,500 kilograms per day pre-atmospheric, which scales to approximately 40,000 tons annually. Micrometeorites make up 99.5% of extraterrestrial material reaching the surface, while larger meteorites contribute only about 8 tons per year. The flux was not constant. Early Earth, during the period some researchers call the Late Heavy Bombardment, a proposed spike in impacts between 4.1 and 3.8 billion years ago, may have experienced infall rates 5 to 6 orders of magnitude higher, though the existence and intensity of that bombardment remains contested. Even without invoking a cataclysm, the baseline delivery over hundreds of millions of years is staggering. Space was not delivering life, it was delivering chemistry, continuously, at a scale that dwarfs any plausible terrestrial synthesis. What arrived in that reign has been catalogued for decades through the study of carbonaceous chondrites, a class of primitive meteorites rich in organic compounds. The Murchison meteorite, which fell near Murchison, Victoria, Australia, on the morning of September 28, 1969, became the reference standard. Over 100 kilograms were recovered across a 35 square kilometre strewn field. Laboratories identified more than 70 amino acids in Murchison, only 8 of which are used by terrestrial biology. The Murchison meteorite also contained purines and pyrimidines, the 2 classes of nitrogenous bases that form the rungs of the DNA and RNA ladders, along with polycyclic aromatic hydrocarbons, sugars, alcohols, and amphiphilic compounds capable of assembling into primitive membrane-like structures. Carbon isotope analysis confirmed the organics were extraterrestrial because the isotopic signatures were inconsistent with terrestrial contamination. Murchison became proof of concept. Space could manufacture and deliver a broad palette of prebiotic molecules. But meteorites, by definition, have touched Earth. The moment a rock strikes the surface, it is exposed to terrestrial biology. Amino acids are everywhere on this planet, in soil, in groundwater, on human hands. Contamination is trivial to introduce and difficult to rule out, even with careful handling. For 50 years, the question lingered. Were the organics in meteorites truly indigenous, or were they terrestrial contamination that had seeped in after the fall? The only way to answer that question was to retrieve a sample that had never touched Earth's surface, never been exposed to its atmosphere, never been handled by gloved hands in a field. The answer required a sample return mission. On December 6, 2020, a capsule released by the Japanese spacecraft Hayabusa 2 streaked through the night sky over Woomera, South Australia, and landed in the desert carrying 5.4 grams of material from the surface of asteroid 162173 Ryugu, a C-type carbonaceous asteroid roughly 900 meters across, orbiting the Sun between Earth and Mars. The sample had been collected in 2019 from 2 touchdown sites on Ryugu's surface and stored in a hermetically sealed container that had never been opened in Earth's atmosphere. Initial analysis, published in 2022 by Yasuhiro Oba and colleagues at Hokkaido University, detected amino acids and the purine nucleobases adenine and guanine. In March 2023, Oba's team published confirmation of uracil, one of the 2 pyrimidine bases unique to RNA. And in March 2026, the full result arrived. All 5 canonical nucleobases, adenine, guanine, cytosine, thymine, and uracil, were present in the Ryugu sample. The complete genetic alphabet, confirmed in material that had never been exposed to terrestrial biology. Oba told reporters the detection strongly supports their ubiquity in the solar system. 3 years after Ryugu's return, on September 24, 2023, NASA's OSIRIS-REx mission delivered 121.6 grams of regolith from asteroid 101955 Bennu, a near-Earth carbonaceous asteroid roughly 500 meters in diameter. The sample mass exceeded the mission's 60-gram requirement by more than a factor of 2, making it the largest asteroid sample ever returned to Earth. The material was processed at NASA's Johnson Space Center in Houston, under strict contamination protocols. On January 29, 2025, 2 papers were published simultaneously in Nature and Nature Astronomy, detailing the sample's chemistry. Daniel Glavin and Jason Dworkin, both at NASA's Goddard Space Flight Center, led the organic analysis. The Bennu sample contained 33 amino acids, including 14 of the 20 used in terrestrial proteins, and 19 non-protein amino acids not found in Earth's biology. Glycine was the most abundant, at 44 nanomoles per gram. The total amino acid concentration was approximately 70 nanomoles per gram. The Bennu sample also contained roughly 10,000 distinct nitrogen-bearing chemical species. Ammonia, at a concentration of 13.6 micromoles per gram, 75 times higher than Ryugu and 12 times higher than Murchison, and a suite of 11 evaporite minerals, including calcite, halite, sylvite, and trona, a sodium sesquicarbonate mineral that had never before been identified in extraterrestrial material. The evaporites implied that liquid water had once been present on Bennu's parent body, and that the water had evaporated under conditions of alkaline pH and temperatures below approximately 55 degrees Celsius. The sample was pristine. Glavin stated at the January 29th briefing, "These organic molecules have all been found previously in meteorites, but the Bennu samples are pristine. So we have a much higher confidence now that these chemical building blocks of life are in fact extraterrestrial in origin and formed in space and are not contaminants from Earth." The contamination question was closed. The delivery question was answered. Carbonaceous asteroids contain amino acids, all 5 nucleobases, sugars, ammonia, phosphates, and the mineral fingerprint of ancient brines. The inventory is real, indigenous, and abundant. Lithopanspermia, in its weakest and most defensible form, that space delivered a large chemical inventory of life's molecular building blocks, is now established science. But the question that follows is not whether the molecules arrived. It is whether their arrival explains the origin of life. Consider the timeline. Earth formed approximately 4.54 billion years ago. The planet's surface solidified, oceans condensed, and by roughly 4.4 billion years ago, possibly earlier, liquid water was stable on the surface. The evidence comes from zircon crystals in the Jack Hills of Western Australia, with oxygen isotope ratios consistent with crystallization in the presence of water. Earth was habitable. Yet the earliest widely accepted fossil evidence of life dates to approximately 3.7 to 3.8 billion years ago, preserved in stromatolite-like structures in the Isua supracrustal belt in Greenland, and graphite inclusions in metamorphosed sediments whose carbon isotope ratios suggest biological fractionation. Some researchers argue for earlier microfossils, candidate structures in 3.5 billion-year-old cherts from the Pilbara Craton in Western Australia, or disputed claims of life as old as 4.1 to 4.28 billion years based on carbon inclusions in ancient zircons, but these remain contested. The conservative estimate is 3.7 to 3.8 billion years. The gap between habitability and life is therefore on the order of 500 to 700 million years, a span longer than the entire Phanerozoic eon, the interval from the Cambrian explosion to the present. If space was continuously delivering the building blocks of life at rates potentially millions of times higher than today, why did life wait 700 million years to begin? The answer, according to the evidence now in hand, is that delivery and assembly are not the same process. Space delivered molecules. It did not deliver life. The distinction becomes sharp when you examine what the molecules themselves can do in the environment where they formed. Amino acids and nucleobases synthesized in the cold vacuum of interstellar space or on the surface of an airless asteroid exist in a chemical state that is thermodynamically stable and biologically inert. They do not react with each other. They do not polymerize into peptides or nucleic acids. They do not spontaneously assemble into protocells. The vacuum of space is an excellent preservative, but a terrible laboratory. What does it mean that the complete genetic alphabet can exist for billions of years in a rubble pile asteroid without ever producing anything resembling life? It means the molecules, by themselves, are insufficient. They are ingredients on a doorstep, not a meal on a table. The meal required a kitchen. That kitchen was Earth. The spark of life was not part of the delivery. It was a terrestrial thermodynamic event. To understand why, return to the chemistry. Amino acids in the Bennu sample, and in most carbonaceous meteorites, are roughly equal mixtures of left-handed and right-handed molecules, a state called Racemic. Life on Earth uses only left-handed amino acids in its proteins and only right-handed sugars in its nucleic acids. This property, called homochirality, is universal across all known biology. For 2 decades, a leading hypothesis held that life inherited its handedness from space, that amino acids delivered by meteorites carried a slight excess of left-handed forms, and that this bias was amplified through subsequent chemistry on Earth. The hypothesis was plausible. Some meteorites, including the Tagish Lake meteorite, which fell in British Columbia on January 18, 2000, show enantiomeric excesses as large as 43 to 59% for aspartic and glutamic acids, with carbon isotopes confirming the excess is indigenous and not contamination. But the Bennu sample contradicted the pattern. Glavin, in an interview published on January 29, 2025, said he was a little disillusioned or disappointed Racemic result because I felt like this invalidated 20 years of research. Dworkin added that the finding throws that theory out. The reason life turned left instead of right, NASA stated, remains a mystery. Keep that result in mind. We will return to it. The delivery mechanism, everyone assumes, delivered a racemic inventory, not a chiral one. Homochirality is likely a terrestrial product. But if the molecules arrived without a handedness bias and without the chemical reactivity needed to assemble into polymers, what changed when they landed on Earth? The answer lies in thermodynamics. Life is a dissipative structure. It exists far from thermodynamic equilibrium and maintains that state by coupling energy-releasing reactions to energy-consuming ones. A cell is not at rest. It is in constant motion, synthesizing molecules uphill against entropy gradients by consuming chemical or light energy downhill. This behavior is not unique to biology. Hurricanes, convection cells, and Bénard patterns are also dissipative structures, though life is the most complex known example. In 2013, Jeremy England, then a physicist at MIT and now an Orthodox rabbi and author, formalized a framework he called dissipation-driven adaptation. The central claim is that matter driven far from equilibrium by an external energy source will, over time, self-organize into configurations that more efficiently dissipate that energy. England argued that the origin and evolution of life follow from the fundamental laws of thermodynamics and, in his words, should be as unsurprising as rocks rolling downhill. The framework does not specify a mechanism. It does not tell you which molecules will self-organize, or where, or how, but it reframes the origin of life question. The question is not whether life violates the second law of thermodynamics, because it does not, but rather which environments on early Earth provided the energy gradients necessary to drive the transition from inert chemistry to self-sustaining replication. Dieter Braun, a biophysicist at Ludwig Maximilian University of Munich, offered a specific answer in 2015. Braun and his colleagues demonstrated that temperature gradients across narrow spaces, heated rock pores for instance, can concentrate dilute molecules by factors of thousands to millions through a process called thermophoresis, also known as the Soret effect. The effect is described by the equation C1 over C0 equals e to the power of minus s sub t times delta t, where C represents concentration, S sub t is the Soret coefficient, and delta T is the temperature difference. In a narrow pore, with one end warmed by volcanic heat and the other cooled by seawater, molecules drift toward the cool end and accumulate exponentially. Brown's experiments showed that long RNA-like polymers, once concentrated, could be selectively amplified through thermal cycling, which melts and reforms base pairs in a process analogous to the polymerase chain reaction used in modern laboratories. Brown stated plainly, life is fundamentally a thermodynamic non-equilibrium phenomenon. His work provided a plausible pathway from dilute molecular soup to concentrated, catalytically active polymers. But concentration alone does not solve the problem. The molecules still need to react. Phosphorus is the bottleneck. Every nucleotide in DNA and RNA contains a phosphate group. Phosphate links the sugars in the nucleic acid backbone. Phosphate is the currency of cellular energy, in the form of adenosine triphosphate. But on early Earth, phosphorus was largely locked in the mineral apatite, calcium phosphate, which is highly insoluble in water and chemically unreactive. Apatite does not donate phosphate groups to organic molecules under plausible prebiotic conditions. The phosphorylation problem, how did early chemistry attach phosphate to sugars and nucleobases, went unsolved for decades. Matthew Pasek, a geochemist at the University of South Florida, identified the solution in meteorites. Schreibersite, an iron-nickel phosphide mineral with the formula FeNi3p, is common in iron and stony iron meteorites, but rare in terrestrial rocks. When schreibersite corrodes in water, it releases phosphite and phosphate ions that are far more reactive than the phosphate in apatite. Pasek estimated that 1 to 10% of the phosphorus in Earth's Hadean crust could have been in the form of schreibersite delivered by meteorites. Laboratory experiments showed that schreibersite can phosphorylate nucleosides, the sugar base units of RNA, in yields of up to 6% at temperatures between 60 and 80 degrees Celsius and pH around 9. The reaction is slow, but it works. Meteoritic phosphorus provided a solution to the phosphorylation problem that terrestrial geology alone could not supply. The cosmic inventory was not inert because the molecules themselves were inert. It was inert because it lacked the thermodynamic engines and reactive elements needed to activate it. Earth supplied both. Two competing hypotheses now dominate the field, each proposing a different terrestrial engine. The first is alkaline hydrothermal vents. The second is hot spring wet-dry cycling. Both invoke non-equilibrium thermodynamics. Both can concentrate and activate the cosmic inventory. Neither has been ruled out. Alkaline hydrothermal vents form where seawater circulates through the oceanic crust, reacts with mantle rocks like olivine and pyroxene in a process called serpentinization, and emerges on the seafloor as warm, alkaline, hydrogen-rich fluids. The reaction produces hydrogen gas, methane, formate, and other small organic molecules, and it raises the pH of the vent fluids to between 9 and 11. The temperature of the fluids ranges from 45 to 90 degrees Celsius, far cooler than black smokers, which exceed 400 degrees and have pH near 2 to 3. Black smokers are too hot and too acidic to support prebiotic chemistry. Alkaline vents are mild. On December 4, 2000, scientists aboard the research vessel Atlantis detected a field of carbonate chimneys rising from the seafloor roughly 2,400 kilometers off the US East Coast, at a depth of about 700 meters on the Atlantis Massif, part of the Mid-Atlantic Ridge. The first crewed dive, using the submersible Alvin, took place the following day, December 5th. They named the site Lost City. The chimneys stand between 30 and 60 meters tall. The tallest, named Poseidon, exceeds 60 meters. The Lost City vents have been active for more than 120,000 years, longer than the existence of anatomically modern humans. Lost City produces hydrogen and methane at concentrations up to 100 times higher than black smokers. The fluids are alkaline, around pH 10. The temperature is moderate. The chemistry is reducing. The environment is stable over geological timescales. Deborah Kelly, a marine geologist at the University of Washington and co-discoverer of Lost City, remarked in 2000 that if this vent field was on land, it would be a national park. In a later interview, she added, laughing, "We have a pretty good general idea of how the systems work, but if you start looking at the details, we don't know very much." The alkaline vent hypothesis, formalized by Michael Russell in the 1990s and developed by William Martin and Nick Lane, holds that life began in the thin mineral walls separating alkaline vent fluids from the acidic ocean. The walls are made of iron sulfide, the same material that forms the catalytic cores of many ancient enzymes. Across those walls, roughly 5 nanometers thick, there exists a natural proton gradient. The ocean is acidic, the vent fluid is alkaline, and the difference in pH translates into an electrochemical potential. The gradient can be quantified using the Nernst equation for proton motive force. Delta P equals delta psi minus the quantity 2.3 RT over F times delta pH, where delta psi is the membrane potential, R is the gas constant, T is temperature, F is Faraday's constant, and delta pH is the pH difference. In modern cells, a proton gradient of roughly 150 to 200 millivolts across a 5 nanometer membrane corresponds to an electric field of approximately 30 million volts per meter. Nick Lane has written that this field strength is equivalent to a bolt of lightning. The vent hypothesis proposes that the first metabolism was not built from scratch by early cells, but inherited from geochemistry. The mineral walls provided the gradient. Early protocells, perhaps simple lipid vesicles enclosing catalytic iron-sulfur clusters, co-opted the gradient by embedding proton pumps in their membranes. Chemiosmosis, the coupling of proton flow to ATP synthesis, is universal in modern life. Bacteria, archaea, and eukaryotes all use it. The universality suggests antiquity. Lane argues that life did not invent chemiosmosis. It discovered it, already running, in the walls of alkaline vents. In 2016, William Martin and Madeline Weiss published a reconstruction of LUCA, the last universal common ancestor of all life on Earth. The reconstruction was based on comparative genomics, tracing 6.1 million protein-coding genes from bacteria and archaea back to 355 protein families that appeared to be present in LUCA. The analysis suggested LUCA was an anaerobic, thermophilic, hydrogen-dependent, carbon dioxide-fixing autotroph that used the Wood-Ljungdahl pathway for carbon fixation and possessed the machinery for nitrogen fixation. The environment implied by the reconstruction was a hydrothermal vent. Martin and colleagues concluded that life likely originated in an alkaline vent setting. But the reconstruction is contested. Critics, including Berkhammer and McGlynn, argue that Weiss and Martin's gene set was not stringent enough, and that as many as 82% of the identified LUCA proteins may not survive stricter phylogenetic criteria. A 2024 paper by Moody and colleagues placed LUCA closer to 4.2 billion years ago, based on molecular clock estimates, which would push the origin of life back near the upper limit of the habitability window. The debate is unresolved, but the thermodynamic core of the vent hypothesis remains. Natural proton gradients across thin mineral walls could have powered the first carbon fixation and driven the emergence of metabolism before genetics. The second hypothesis is hot spring wet-dry cycling. In 2015 and 2020, Bruce Damer and David Damer, working at the University of California, Santa Cruz, proposed that life began not in the ocean, but on land, in freshwater volcanic pools that underwent repeated cycles of wetting and drying. The idea draws on laboratory experiments showing that lipid vesicles, spherical bilayer membranes resembling cell membranes, can spontaneously form when lipid molecules are dried onto a surface and then rehydrated. During the drying phase, the lipids and other organics are concentrated. During the rehydration phase, they assemble into membranes and encapsulate whatever molecules are nearby, including potential genetic polymers. Repeated cycling can drive polymerization of RNA-like molecules in a process analogous to the thermal cycling used in PCR. Damer and Damer argue that volcanic hot springs on early Earth's landmasses provided the necessary conditions: fresh water, a source of heat, repeated wet-dry cycles, and exposure to ultraviolet light, which could have driven photochemical reactions. Geological evidence for early hot springs exists. The 3.48 billion-year-old Dresser Formation in Western Australia contains geyserite, a silica deposit characteristic of hot spring systems, in layers interbedded with stromatolites, suggesting that microbial mats colonized hot spring environments very early. The hypothesis is testable in principle and has generated laboratory support, but it faces challenges. Fresh water on early Earth was likely dilute compared to the concentrated brines that formed in tidal pools or salt flats. UV exposure, while driving some useful chemistry, also damages nucleic acids. And the hypothesis requires that the first life emerged on land, not in the ocean, which places it in direct competition with marine origin scenarios like the alkaline vent hypothesis. A third line of research, led by John Sutherland and Matthew Powner at the University of Cambridge, has demonstrated high-yield synthetic pathways to ribonucleotides, the monomers of RNA, from simple precursors like hydrogen cyanide, formaldehyde, and inorganic phosphate, under conditions that might have existed on early Earth's surface. In 2009, Powner and Sutherland published a synthesis of activated pyrimidine ribonucleotides using ultraviolet-driven chemistry in the presence of cyanide and sulfite. The reaction bypassed the traditional bottleneck of ribose instability by building the sugar and base together in a single pathway, rather than attempting to join them after the fact. Subsequent work showed that similar chemistry could produce amino acids and lipid precursors from a shared feedstock, suggesting that life's major building blocks could have emerged through a unified proto-metabolic network, rather than separate, unrelated pathways. The synthesis requires specific geochemical conditions, including sulfite, which could have been supplied by volcanic sulfur dioxide dissolving in water, and ultraviolet light, which was abundant on an early Earth without an ozone layer. Sutherland's work does not specify a single environment, but rather a plausible surface chemistry that could have operated in impact-generated craters, volcanic settings, or tidal flats. The strength of the approach is its demonstrated chemistry. The weakness is that it has not yet been extended to full autocatalytic closure, the point at which the system can sustain and replicate itself without external input. Gunther Wechtershäuser, a German chemist and patent lawyer, proposed in 1992 an alternative framework called the iron-sulfur world. Wechtershäuser argued that life began not with genetics, but with metabolism, specifically with catalytic reactions on the surface of iron sulfide minerals like pyrite. The surfaces could have concentrated reactants, stabilized intermediates, and catalyzed carbon fixation through a primitive version of the reductive citric acid cycle. Iron-sulfur clusters are ubiquitous in ancient enzymes, including those involved in electron transport and carbon fixation, and their presence across all domains of life suggests deep evolutionary antiquity. Laboratory work by Martina Preiner, Jessica Varma, Srijith Varma, Natalia Munyabak, Joseph Moran, Verena Soyo, Sean McGlynn and others has shown that iron-promoted reactions can drive synthesis of amino acids and small organic molecules under conditions resembling those in hydrothermal systems. But the iron-sulfur world has not produced a demonstrated pathway to self-replication, and it remains an influential but incomplete framework. What does it mean that multiple plausible terrestrial engines exist, each with laboratory support and geological analogues, but none yet proven? It means the question of which engine ignited life is genuinely open. The evidence does not yet favor one over the others. But the evidence does favor the general conclusion. Life required a terrestrial thermodynamic engine to activate the inert cosmic inventory. Space delivered the molecules, Earth provided the conditions under which those molecules could concentrate, react, polymerize, and eventually close into autocatalytic cycles capable of self-replication. Panspermia, in its pseudo form, is part of the answer, but it is not the whole answer. The spark was local. The Racemic Bennu result returns now with sharper focus. If homochirality was not inherited from space, it must have emerged on Earth. How? The question has a mathematical structure. Chirality is described by enantiomeric excess, EE, equal to the absolute value of L minus D divided by L plus D times 100, where L and D represent the concentrations of left-handed and right-handed forms. A racemic mixture has EE equal to 0. Pure homochirality has EE equal to 100. The transition from 0 to 100 is a symmetry-breaking event. In 1953, F.C. Frank proposed a mechanism. If a chiral molecule can catalyze its own formation and simultaneously inhibit the formation of its mirror image, the system will spontaneously amplify any initial small excess into near-complete homochirality. The mathematics of the model involve coupled differential equations describing autocatalysis and mutual antagonism. Frank's model was abstract, but in 1995, Kenso Soai, a chemist at the Tokyo University of Science, discovered a real chemical system that behaves exactly as Frank predicted. The Soai reaction, involving asymmetric autocatalysis of chiral zinc alkoxides, can amplify an initial enantiomeric excess of 2% to greater than 99.5% through repeated catalytic cycles. The amplification is exponential and requires only that the reaction couple product formation to chiral bias. Soai's discovery, published in Nature in 1995, demonstrated that homochirality can emerge from a nearly racemic starting point through chemistry alone, without invoking external templates or selection. If early Earth provided environments where chiral autocatalysis could operate, perhaps in lipid vesicles where concentrated organics underwent repeated reaction cycles, or on mineral surfaces that selectively adsorbed one enantiomer, then a small initial fluctuation, perhaps introduced by circularly polarized light from a neutron star or reflected off atmospheric ice crystals, could have been amplified into the universal handedness observed in all life today. The Tagish Lake meteorites' large left-handed excess demonstrates that such biases can exist in extraterrestrial material. But the Racemic Bennu sample demonstrates that they need not. Homochirality is likely terrestrial, amplified through Soai-type autocatalysis, not inherited from a pre-existing cosmic bias. The ingredients were cosmic. The concentration, activation, polymerization, and chiral amplification were terrestrial. The spark was not delivered. It was ignited here, in environments where energy gradients, thermal, chemical, electrochemical, could drive matter far from equilibrium and hold it there long enough for autocatalytic closure to occur. But the timeline remains a puzzle. If Earth was habitable by 4.4 billion years ago, if the cosmic inventory was being delivered at rates potentially millions of times higher than today, If thermodynamic engines like alkaline vents or hot springs existed from early in Earth's history, why did life take 500 to 700 million years to begin? One answer is that the rock record before 3.8 billion years ago is almost entirely missing. The crust was recycled through subduction and metamorphism. The oldest surviving crustal rocks are the 4.4 billion-year-old zircons from the Jack Hills, which are single mineral grains preserved in much younger sediments, not intact rock formations. The Isua supracrustal belt in Greenland, dated to 3.7 to 3.8 billion years, is among the oldest surviving rock sequences, and it is heavily metamorphosed. Anything that happened before 3.8 billion years ago, including possibly the origin of life itself, may have left no geological trace. The gap in the timeline may be a gap in the record, not a gap in the process. Life could have started earlier, we simply cannot see it. Another answer is that autocatalytic closure is hard. The transition from a mixture of reactive organic molecules to a self-sustaining, self-replicating system capable of Darwinian evolution is not automatic. It requires that the system close, that the products of the reaction network include the catalysts needed to sustain the network. Closure is a threshold. Below the threshold, the system dissipates. Above it, the system persists and evolves. Laboratory efforts to construct minimal self-replicating systems from scratch have not yet succeeded, despite decades of work. The difficulty suggests that the transition, while not violating any physical law, is a low-probability event, dependent on specific chemical and environmental conditions. If the probability is low, then a 500 million year waiting time is not surprising. It may even be fast. The question shifts. Is autocatalytic closure inevitable given the right conditions? Or is it a contingent event whose occurrence on Earth was, in some sense, fortunate? The evidence does not yet settle that question, but it does rule out one tempting answer: that the answer is simple. England's framework does not specify which molecules will self-organize, only that matter driven far from equilibrium will, over time, find configurations that dissipate energy more efficiently. The framework applies to hurricanes, to convection cells, to the formation of stars, and to the origin of life. In an interview, England called origin of life questions a fraught mess based on fragmentary evidence he was inclined to steer clear of for now. His caution is warranted. The evidence is fragmentary. But his broader claim stands. The origin and evolution of life follow from fundamental thermodynamic laws and should be, in his words, as unsurprising as rocks rolling downhill. Life is not a violation of physics. It is an expression of it, occurring wherever the conditions permit. The cosmic inventory of building blocks delivered by asteroids and comets is one of those conditions. Terrestrial thermodynamic engines are another. Together, they are sufficient to explain the origin of life on Earth, without invoking either a verdict one way or the other on the ultimate origin. The science resolves the mechanism. The metaphysical question, why the laws of thermodynamics permit dissipative structures to exist at all, why there is something rather than nothing, is not answered by the mechanism. It is beyond what the measurement can settle, and it is honest to say so. Before I continue, consider what this means. Take a moment. Space delivered every molecular building block needed for life, and delivered them in quantities that dwarf any plausible terrestrial synthesis. Yet those molecules, sitting inert in a vacuum for billions of years, never assembled into anything resembling life. The spark required thermodynamic engines that existed only on Earth. What does it mean that the complete genetic alphabet can exist in a lifeless asteroid? It means the alphabet is not the sentence. The spark is not the delivery. The origin of life is a thermodynamic event, not a chemical inevitability. Return now to the two competing terrestrial hypotheses and ask which one the evidence supports. The alkaline vent hypothesis has several strengths. Lost City is a real, long-lived analogue that has been producing hydrogen, methane, and organic molecules continuously for more than 120,000 years. Chemiosmosis, the coupling of proton flow across a membrane to the synthesis of ATP, is universal across every domain of life on Earth. Bacteria use it, archaea use it, eukaryotes use it. When a feature is universal, the most parsimonious explanation is that it was present in the common ancestor of all living things. And chemiosmosis is not a trivial feature. It is a complex, membrane-embedded machinery involving proton pumps, rotary ATP synthase enzymes, and precisely tuned electrochemical gradients. The odds that such a system evolved independently multiple times are vanishingly small. The most likely explanation is that chemiosmosis is ancient as Lucca itself, and that its universality points to an origin in an environment where proton gradients already existed, ready to be co-opted. Alkaline hydrothermal vents provide exactly such an environment. The proton gradient across the thin iron sulfide walls of a vent, quantified by the Nernst equation at a field strength comparable to a bolt of lightning, is a free geochemical gradient that the first protocells could have harnessed before they possessed the genetic machinery to build their own. This is the deepest strength of the vent hypothesis. It does not require life to invent the hardest thing life does. It lets geochemistry provide it. Think about a locksmith who inherits a workshop. The tools are already on the wall, already sharpened, already arranged. The locksmith does not forge the tools from raw ore before beginning work. He picks up what is there and uses it. The alkaline vent hypothesis casts early life as that locksmith. The proton gradient was already on the wall, generated by the geology of serpentinization, maintained across the mineral membrane by the pH difference between alkaline vent fluid and acidic ocean. Life did not forge chemiosmosis from nothing. It inherited a running gradient and learned to tap it. When you consider that every cell in your body at this moment is maintaining a proton gradient across its mitochondrial membranes at roughly 150 to 200 millivolts, a voltage per nanometer that rivals a lightning strike, and that this same mechanism powers every organism from the deepest thermophile to the tallest sequoia, the hypothesis that the mechanism was inherited from geology rather than invented by biology carries real weight. But the alkaline vent hypothesis has weaknesses, and honesty requires naming them. The mineral walls of a vent are thick, micrometers across, not the 5 nanometers of a cell membrane. A proton gradient across a thick inorganic wall does not translate cleanly into a gradient across a thin biological membrane. The transition from a geochemical gradient in a mineral structure to a biological gradient in a lipid bilayer is not chemically trivial. And critics argue that the vent hypothesis has not fully explained how it occurred. There is also the dilution problem. The open ocean is an enormous volume of water, and any organic molecules synthesized at a vent would be immediately diluted into that volume, dispersing before they could concentrate to reaction-relevant levels. Vent proponents counter that the porous mineral structure of the chimney itself provides confined spaces, microcompartments where molecules could concentrate. But the adequacy of that confinement is disputed. The vent hypothesis is the leading marine origin scenario, supported by the universality of chemiosmosis, the reconstruction of Lucca as a hydrogen-dependent thermophile, and the existence of Lost City as a working analogue. But it is not proven, and its weaknesses are real. The hot spring hypothesis of Bruce Damer and David Deamer inverts nearly every assumption of the vent hypothesis. Where the vent hypothesis places life's origin in the deep ocean, the hot spring hypothesis places it on land. Where the vent hypothesis emphasizes metabolism-first chemistry, powered by proton gradients, the hot spring hypothesis emphasizes membrane and RNA-first assembly powered by wet-dry cycling. The core insight of Damer and Deamer's work is that concentration and polymerization are difficult in bulk water. Water hydrolyzes bonds. It pulls polymers apart. To build long chains of RNA or peptides, you need to remove water at some stage. You need a drying phase. Freshwater volcanic pools on early Earth's landmasses would have undergone repeated cycles of filling and evaporation, driven by rainfall, geothermal heat, and the daily and seasonal rhythms of the environment. During the drying phase, organic molecules concentrate at the edges of the pool, and lipid molecules organize into stacked multilamellar structures, sheets of membrane pressed together, with organic molecules trapped between the layers. In that concentrated, water-poor environment, polymerization can occur. Nucleotides can link into short RNA strands, amino acids into short peptides. During the subsequent wetting phase, the membranes rehydrate and bud off into vesicles, each one encapsulating whatever polymers formed during drying. Repeated over thousands of cycles, this process could progressively select for longer, more stable polymers and package them inside protective membranes, the beginnings of a protocell. Deamer's laboratory has demonstrated key steps of this process experimentally. When mixtures of nucleotides and lipids are subjected to repeated wet-dry cycles at temperatures resembling a hot spring, RNA-like polymers up to 100 nucleotides long can form, encapsulated within lipid vesicles. The chemistry works in the lab, and there is geological support. The Dresser Formation in the Pilbara region of Western Australia, dated to approximately 3.48 billion years ago, contains geyserite, a distinctive silica deposit that forms only around hot springs and geysers, interbedded with stromatolites, the layered structures built by microbial communities. The presence of geyserite, bedded with evidence of early microbial life, demonstrates that microbes colonized hot spring environments very early in Earth's history. It does not prove life originated there, but it establishes that the environment existed and was hospitable to life at a time close to life's origin. Picture a tidal flat at the edge of a volcanic lake, the water rising and falling with each rain and each dry spell. At the waterline, a ring of concentrated organic residue accumulates, a bathtub ring of prebiotic chemistry. Each time the water recedes, the ring dries, and molecules pressed together in the absence of water link into chains. Each time the water returns, the chains float free, packaged inside newly formed membrane bubbles. Over 10,000 cycles, a span of perhaps a few decades or centuries, the pool becomes a natural combinatorial laboratory, testing countless molecular arrangements, preserving the stable ones, discarding the rest. This is the image the hot spring hypothesis offers. Not a single miraculous event, but a relentless cyclic process. A chemical engine driven by the sun and the rain and the volcanic heat beneath. But the hot spring hypothesis faces its own difficulties. Fresh water is dilute. Unlike the confined pores of a vent chimney or the concentrated brines of a tidal salt flat, an open freshwater pool contains molecules at very low concentrations, and the wet-dry cycling must overcome that dilution before any useful chemistry can occur. Ultraviolet light, abundant on an early Earth without an ozone layer, is a double-edged tool. It can drive useful photochemistry, as Sutherland's work demonstrates, but it also damages nucleic acids, breaking the very bonds the process is trying to form. And the hot spring hypothesis places life's origin on land, in competition with marine scenarios, at a time when Earth's land masses were limited. The early Earth may have been largely or entirely covered by ocean, with few exposed volcanic islands to host freshwater pools. The hypothesis is testable, laboratory-supported, and geologically plausible. But it is not proven, and its assumptions, freshwater, land, membrane first, RNA first, are each contestable. Weigh the two against each other, and a striking pattern emerges. They are not obviously distinguishable on the current evidence. Both invoke non-equilibrium thermodynamics. Both concentrate dilute molecules through physical gradients. The vent through proton and thermal gradients across mineral walls. The hot spring through wet-dry cycling. Both can, in principle, activate the inert cosmic inventory. The vent hypothesis has the advantage of explaining chemiosmosis, the universal energy currency of life, and of aligning with the LUCA reconstruction as a hydrogen-dependent thermophile. The hot spring hypothesis has the advantage of solving the polymerization problem through drying, and of demonstrated laboratory synthesis of encapsulated RNA. The vent hypothesis struggles with the transition from thick mineral wall to thin biological membrane. The hot spring hypothesis struggles with dilution and UV damage. Each explains what the other cannot. Neither explains everything. This is not a failure of science. It is the honest state of a frontier where the evidence is genuinely balanced, and the experiments that would decide the question cannot yet be run. Consider the fundamental obstacle. nobody can run a 100 million year experiment. The origin of life, on any hypothesis, unfolded over a span of time that dwarfs not only human history, but the entire existence of our species. A laboratory experiment runs for weeks, months, at most years. It can demonstrate individual steps, the synthesis of a ribonucleotide, the polymerization of a short RNA strand, the formation of a lipid vesicle, the amplification of a chiral excess, but it cannot compress 100 million years of continuous cycling into a fundable research program. This is the deepest methodological limit in origin of life science. There is no fossil of the transition from non-life to life. The first cell left no skeleton. The chemistry that preceded it left no trace in the rock record, which in any case does not survive from before 3.8 billion years ago. Scientists are reconstructing a singular event that happened once, roughly 4 billion years ago, in an environment they cannot directly observe. From chemistry, they can only partially replicate. That they have narrowed the possibilities to a handful of plausible engines, each grounded in demonstrated chemistry and geological analogue, is a genuine achievement. That they cannot yet name the winner is not a weakness. It is the honest boundary of what the evidence currently permits. Think about a forensic investigator arriving at a scene long after the event. The investigator cannot watch the event happen. Instead, she reconstructs it from what remains: the position of objects, the chemistry of residues, the pattern of traces. If two reconstructions both fit the available evidence equally well, the honest investigator does not declare a verdict she cannot support. She states which reconstructions the evidence permits, which it rules out, and where the evidence is silent. Origin of life science is forensic in exactly this sense. The vent hypothesis and the hot spring hypothesis both fit the surviving evidence. The pure panspermia hypothesis, that life itself, not just its molecules, arrived from space, does not fit, because it merely relocates the problem without solving it, and because there is no evidence of viable organisms surviving interstellar transit and successful delivery. The radiopanspermia of Arrhenius is ruled out. Spores driven by radiation pressure would be sterilized by ultraviolet light and cosmic radiation over the millions of years such a journey would require. The directed panspermia of Crick and Orgel is unfalsifiable and therefore outside the reach of scientific evidence. What survives forensic scrutiny is pseudo-panspermia, the delivery of molecules coupled to a terrestrial thermodynamic engine whose specific identity remains open. There is one more thread to draw in, and it concerns the resilience of life once it exists. The tardigrade, a microscopic 8-legged animal less than 1 millimeter long, is the most durable known organism. Tardigrades can survive desiccation, freezing to near absolute zero, pressures greater than those in the deepest ocean trenches, and radiation doses that would kill a human many times over. The median lethal dose of gamma radiation for a tardigrade is approximately 5,000 grays, and for heavy ion radiation, approximately 6,200 grays. For comparison, a dose of 5 to 10 grays is fatal to a human. In September 2007, Aboard the European Space Agency's Photon M3 mission, tardigrades became the first animals known to survive direct exposure to the raw vacuum of space combined with solar ultraviolet radiation in low Earth orbit at an altitude of 258 to 281 kilometers. Some individuals not only survived, but subsequently reproduced. The tardigrade's resilience is often cited in support of panspermia. If an animal can survive space vacuum and radiation, perhaps organisms could survive interstellar transit. But the tardigrade experiment cuts the other way when examined carefully. The tardigrades survived hours to days in low Earth orbit, protected by the relatively short exposure and by their own remarkable biochemistry, which includes proteins that vitrify their cells into a glass-like state during desiccation. Interstellar transit takes not hours, but millions of years, exposing any organism to a cumulative radiation dose vastly beyond even a tardigrade's tolerance. The tardigrade demonstrates that life, once it exists, can be astonishingly durable. It does not demonstrate that life can originate in space or survive the journey between stars. The distinction matters. Resilience is not origin. An organism that can endure a brief exposure to vacuum is not evidence that life assembled itself in the cold and dark and rode intact to a new world. It is evidence only that biology, once established, can armor itself against extremes. What does it mean that the most durable organism we know cannot survive the journey that pure panspermia requires? It means the burden of the origin of life falls back onto chemistry, onto the delivered molecular inventory and the terrestrial engines that activated it. The tardigrade, for all its toughness, does not carry life across the galaxy. The molecules do the traveling. And the molecules, as the pristine Ryugu and Bennu samples confirmed, travel inert. Return once more to the numbers, because the numbers discipline the story. The Bennu sample contained 33 amino acids, including 14 of the 20 used in terrestrial proteins. It contained all classes of the building blocks. It contained ammonia at 13.6 micromoles per gram, a nitrogen source essential for building amino acids and nucleobases. It contained a complete set of evaporite minerals, the chemical signature of an ancient brine that had once been liquid and then dried. Direct evidence that Bennu's parent body once hosted flowing alkaline water at temperatures below 55 degrees Celsius. The trona in the Bennu sample, a sodium sesquicarbonate never before seen in extraterrestrial material, forms specifically through the evaporation of sodium carbonate-rich water. Bennu's parent body was, at some point in the early solar system, a place with liquid water and active aqueous chemistry. And yet, despite liquid water, despite nitrogen, despite amino acids and nucleobases and phosphates, all present together, Bennu's parent body did not produce life. The ingredients were present, and the water was present, but the parent body was too small, too cold, too short-lived in its wet phase to sustain the continuous, driven, non-equilibrium chemistry that the origin of life requires. What does it mean that an asteroid can have liquid water and all the building blocks and still produce nothing living? It means water and molecules are necessary but not sufficient.
The origin of life requires more: sustained energy gradients, confinement, cycling, and time. The specific thermodynamic conditions that a small asteroid cannot maintain, but a young planet can. This is the part that stays with me. Not the presence of the molecules in space, remarkable as that is. The absence of life, despite their presence. Bennu had liquid water. Bennu had amino acids and all 5 nucleobases in its parent body. Bennu had ammonia and phosphates. Bennu had everything on the ingredient list. And Bennu is a dead rock. The gap between having the ingredients and being alive is the entire mystery. And the pristine samples make that gap visible in a way no meteorite study ever could. Because a meteorite that has sat in a field could always be dismissed as contaminated. The pristine samples cannot be dismissed. They show, with confidence, that the cosmos routinely assembles the parts of life and routinely fails to assemble life. The parts are common. The assembly is not. The phosphorus story deserves one more turn, because it illustrates the general principle with precision. On modern Earth, phosphorus cycles through the biosphere in vast quantities, moving from rock to soil to organism and back. But on the early Earth, before life had begun to actively mobilize it, phosphorus was chemically imprisoned. The dominant phosphorus mineral, apatite, is calcium phosphate, and it is one of the least soluble common minerals
in Earth's crust. Its phosphate groups do not readily transfer to organic molecules. If early Earth's only phosphorus source had been apatite, the phosphorylation of nucleosides, the attachment of phosphate groups to build the RNA backbone, might have been impossibly slow. The solution, identified by Matthew Pasek, came from the same source as the organic inventory: space. Meteoritic schreibosite, the iron-nickel phosphide with formula FeNi3p, is far more reactive than apatite. When schreibosite corrodes in water, it releases reduced phosphorus species, phosphite and hypophosphite, that are soluble and chemically active. These reduced phosphorus compounds can phosphorylate nucleosides in aqueous conditions, achieving yields of up to 6% at 60 to 80 degrees Celsius and alkaline pH. Pasek estimated that between 1 and 10% of the phosphorus in Earth's Hadean crust may have arrived as schreibosite in meteorites. Consider the elegance of this. The same cosmic delivery that brought the amino acids and nucleobases also brought a reactive form of phosphorus that terrestrial geology could not supply. Space did not deliver life, but it delivered more than the carbon-based building blocks. It delivered the reactive phosphorus that made those building blocks assemblable. The cosmic contribution was real and specific. Not the spark itself, but the fuel and much of the raw material the spark would consume. Think about an accountant reconciling 2 ledgers. The first ledger, the cosmic one, records everything space delivered: amino acids, nucleobases, sugars, ammonia, phosphates, reactive schreibosite phosphorus, amphiphilic molecules capable of forming membranes. The second ledger, the terrestrial one, records what Earth added: sustained thermal gradients in rock pores, proton gradients across mineral walls, wet-dry cycling in volcanic pools, ultraviolet-driven surface chemistry, and the deep, uninterrupted time that a planet can offer and an asteroid cannot. When you reconcile the two ledgers, the origin of life balances only when both are counted. Neither ledger alone produces life. The cosmic ledger alone gives you Bennu, a dead rock full of ingredients. The terrestrial ledger alone gives you a planet with energy gradients, but without the delivered inventory, a much thinner supply of the specific molecules life requires. Life required both columns. The spark required the terrestrial column specifically, but the fuel it burned came substantially from the cosmic one. The Gibbs free energy equation captures the thermodynamic heart of the matter. The change in free energy of a reaction, delta G, equals delta H minus T times delta S, where delta H is the change in enthalpy or heat content, T is the absolute temperature, and delta S is the change in entropy or disorder. A reaction proceeds spontaneously only when delta G is negative. Many of the reactions that build life's polymers, linking amino acids into peptides, nucleotides into nucleic acids, have positive delta G. They are not spontaneous. They do not happen on their own. Life solves this by coupling. It drives an unfavorable reaction with positive delta G by linking it to a favorable reaction with a larger negative delta G, so that the sum is negative and the coupled process proceeds. This coupling is the essence of metabolism. But before enzymes existed to orchestrate the coupling, the early Earth had to supply the energy some other way, through thermal gradients, through proton gradients, through the removal of water during drying, which shifts the equilibrium of condensation reactions toward polymerization. Every candidate terrestrial engine is, at its core, a way of supplying free energy to push unfavorable reactions forward. The alkaline vent supplies it through a proton gradient. The hot spring supplies it through wet-dry cycling that removes water and drives condensation. The thermal pore supplies it through concentration. Each engine is a different answer to the same thermodynamic question. How do you make positive delta G reactions happen without enzymes? The molecules from space could not answer that question in a vacuum. Only a driven, non-equilibrium terrestrial environment could. What would you conclude from this evidence? Consider the full weight of it before answering. Space delivers the molecular inventory, confirmed pristine, confirmed abundant, confirmed ubiquitous across the solar system. The inventory is racemic, so handedness was not inherited from space. The inventory is inert, so it does not self-assemble in a vacuum. Reactive phosphorus arrives alongside the organics, solving the phosphorylation bottleneck. Multiple terrestrial thermodynamic engines exist, each capable of concentrating and activating the inventory, each grounded in demonstrated chemistry and geological analog. Chiral autocatalysis, demonstrated in the Soai reaction, can amplify a small excess into homochirality without a cosmic template. A 500 to 700 million year gap separates habitability from the earliest accepted life. A gap that may reflect either a missing rock record or the genuine difficulty of achieving autocatalytic closure. Put these together, and the conclusion the evidence supports is clear in its outline and honest in its limits. Life's chemical building blocks arrived from space. Life itself did not. The spark was a local terrestrial thermodynamic event, the activation of an inert cosmic inventory by energy gradients that only a young planet could sustain. Space wrote the alphabet, Earth composed the first sentence. But which sentence exactly? And in which room of the house it was composed? The deep sea vent or the volcanic pool? The evidence does not yet say. And there is a further edge, deeper than the choice of engine. Even if science identifies the precise environment and the precise chemistry, even if a laboratory someday demonstrates full autocatalytic closure under vent or hot spring conditions, that achievement would explain the mechanism by which inert chemistry became living chemistry. It would not explain why the universe is the kind of place where thermodynamics permits dissipative structures to arise at all. Why the laws are such that matter driven from equilibrium can organize, persist, and eventually reflect on its own origin. That question stands at the boundary of what physical measurement can reach. The science describes how. The question of why there is a something in which how can happen is not a scientific question. And the honest position is to name that boundary plainly and leave it standing, closing it neither one way nor the other. The equations resolve the mechanism. They do not resolve the ultimate origin, and they do not pretend to. I return to this point often, not because it is comfortable, but because it is true. The most rigorous science in the world can trace the pathway from a molecule in a cold asteroid to a self-replicating system in a warm terrestrial pore. And that tracing is one of the great intellectual achievements of our species. But at the end of that pathway, when the mechanism is fully described, a question remains that the mechanism cannot touch. Science answers the question it can measure. It reaches the edge of measurement and stops. What lies beyond that edge is not answered by the vent hypothesis or the hot spring hypothesis or the thermodynamics of dissipation. It is left open. And there is a particular kind of honesty in a discipline that can say, with confidence, exactly how far its methods reach and exactly where they stop. Let me bring this exploration to a close. We began with panspermia, the hypothesis that life or its building blocks rode to Earth aboard space rocks, and with the two pristine samples, 5.4 grams from Ryugu, 121.6 grams from Bennu, together less than a small apple, that confirmed what 50 years of meteorite studies could only suggest. That the complete genetic alphabet, all 5 nucleobases, along with amino acids, sugars, ammonia, and reactive phosphorus, exists in lifeless carbonaceous asteroids and was delivered to the early Earth in staggering quantities. We traced the delivery from Arrhenius's radio panspermia